This research project was designed to investigate the effect of saffron extract on dopamine release from Schreiber cells. Neurotransmitters are chemicals in our body that allow neurons to communicate with each other. There are different types of neurotransmitters, such as dopamine, serotonin, and glutamate. Dopamine is one of the major neurotransmitters in our body that has physical effects such as motor functioning, as well as psychological effects such as regulating mood. Moreover, serotonin is another major neurotransmitter, most commonly known for having direct impact on mood, sleep, and digestion. Saffron is the most expensive spice in the world because harvesting the flower and separating the stigmas from the flower is done by hand. Several studies have been shown that saffron can increase the production of dopamine, serotonin, and glutamate in our body. It is hypothesized that the saffron increases the dopamine release in Schreiber cells. Therefore, the aims of the study are to establish a UPLC method as a substitute for ELISA to quantify dopamine release of the cells and treat them with various concentrations of saffron to determine its effect on dopamine release levels. An increase in dopamine and serotonin receptor gene expression could represent an increase in dopamine and serotonin levels. Reverse transcription PCR or RT-PCR evaluates this after saffron treatment. For my method, I started plating the Schreiber cells and then treat them with two different doses of saffron, 50 and 100 micromolar. My method includes two parts, a UPLC section and a PCR section. For my UPLC part, I first started my method validation where I tested different columns, injection volumes, flow rate, detection limit, and mobile phase type and gradient using a dopamine standard. Then, with validated parameters, I tested the collected supernatant and compared it to the live sample, and used that information to detect dopamine in all of my samples. For my PCR part, I extracted the RNA from the treated cells and reverse transcribed it to cDNA. Figure 1 shows the morphological changes of Schreiber cells before and after treatment, where A is a representative of pre-treatment cells, B is a representative of untreated cells after 24 hours, and C shows the cells after the saffron treatment at that time point. As shown here, no morphological changes were observed. Then, as shown here in figure 2, I was able to quantify dopamine using amide UPLC column while having an internal standard. First, I compared the supernatant dopamine level to lyse dopamine level. Since lyse sample had one biological replicate, no statistical analysis were performed, but as shown on the graph, it appeared to contain more dopamine and was used afterwards. Then the dopamine was quantified in treated cells. Data was underpowered for statistical analysis and further experiments are needed. Figure here shows the gene expression of dopamine and serotonin receptors with 3 biological replicates and 2 technical replicates, where GAPTH was used as the housekeeping gene. I used 2 dopamine receptors gene, D1 and D2, and 2 serotonin receptors gene, HTR1B and HTR2A. 2-way ANOVA test was performed and no significant differences were observed. To conclude, a UPLC method was established to quantify dopamine content from the treated cells. Also, PCR assessed the gene expression of dopamine and serotonin receptor after treating Schreiber cells with saffron, and no significant differences were observed. At the end, I would like to thank my instructors and my classmates for their support and patience throughout this research and this program. Thank you.